Hello, Sridhar sir. Am I audible?
Yeah.
Okay. We'll start the session. Eminent speaker of today's session, Dr. R. Amarendra Varma Garu, postdoctoral researcher, Germany. Dr. K. Srinivasulu Garu, Head of Biotechnology Department, KL University. Dr. Jahurullah Muhammad Garu, Chief Innovation Officer, Government of India. Participants from various colleges and universities, a very good afternoon to one and all. Welcome to third day of a 4-day international webinar, BioConclave, on an intellectual war against COVID-19 pandemic, organized by Department of Pharmaceutical Biotechnology, Hindu College of Pharmacy, Guntur. I hope you acquire more information on COVID-19 evolution and pathogenicity from yesterday's session. Today's speaker, Dr. R. Amarendra Varma Garu, can deliver a lecture on host T cells and microRNA role in COVID-19. I want to give a brief outline regarding today's topic, that is host T cells and microRNA role in COVID-19. MicroRNAs are a class of conserved sequences, 20 to 23 nucleotides long, single-stranded small RNAs that inhibit expression of their respective target genes in different cell types. Regulatory T cells are a subpopulation of T cells that negatively regulate immune responses, which is essential to immune homeostasis. There has been evidence that microRNAs are involving in many cellular processes such as proliferation, differentiation, and apoptosis. Their dysregulation could lead to many diseases such as cancer, cardiovascular diseases, and diabetes. A growing body of recent research has indicated that microRNAs play an important role in controlling the development and function of immune cell types. If we have the knowledge on host T cells, T cell regulation, we can understand the mechanisms of immune disorders. Today's speaker, Amar Varma sir, has a vast research experience in immunology. With this brief introduction, I am closing my opening remarks. Now I request the co-convener of today's program, Dr. P. Jawahar Babu sir, to introduce today's speaker. Over to Jawahar Babu sir. Sir.
Good afternoon to one and all.
Jawahar Babu.
Yeah, am I audible?
Yeah. You okay? First to give the share rights to the Amar Varma. Share screen.
Yeah.
Then I will give how to stop
Sir?
that.
Yeah.
Hello.
Yeah.
Sir, share screen option is given, sir.
Given, okay.
Hmm.
Amar. Okay. So right side, 3 dots are there. Amar, are you getting my voice?
Yeah.
Yeah, where actually right side? Share screen.
Yeah, share screen and then
options is there. Side, right side.
1 minute. Options.
3 dots will appear. 3 dots.
More, sir.
More options, sir.
Yeah, more options. Yeah.
Okay. Yeah, right here. More options. Yeah,
where you will find disable
annotations.
Disable participants annotation.
Yeah.
Right.
That's enough.
Yeah.
Remaining thing you can do.
Okay.
Yeah. Jawahar.
So, yeah, it's now. Is it okay?
Jawahar.
Yes, sir.
Yeah. Due to busy schedule, I may leave the meeting at 3 or 3:10. You assist for you give your support to the Amar Varma.
Yeah.
During the
sir.
question session and other.
Yes, sir. Definitely.
Yeah. Because today is some eclipse, na?
Okay.
Solar eclipse. That's why. Okay.
Yeah.
You continue.
On behalf of our college, Chairman Sir, Sri Dupudi Rangaraj Garu, our Secretary and Correspondent Sir, Sri Madhusudan Rao Garu, teaching faculty, participants of this international webinar. I feel that it is an immense pleasure to welcome and introduce Dr. R. Amar Varma Garu. He is an intellectual, versatile personality in the field of immunology and pharmaceutical biotechnology. He is an intellectual person in respect of his research experience. Dr. Amar Varma Garu completed his master degree from Maynooth University, Ireland, and doctorate in immunology from OU Pottango Gucci University, Germany. He has done many research projects on human T cells, focusing on their biology, function, and role. He worked as a guest scientist and has contributed research to the Institute of Molecular and Clinical Immunology in Germany multiple times. Sir, his expertise immunology, molecular marker, gene expression profiling by quantitative PCR, and pharmaceutical biotechnology. Sir has published his research work in various reputed high-impact peer-reviewed journals, including high-impact science signaling. It is around 7.0 impact factor. Class 1, it is around 3 impact factor, and cell communication signal. All are highly impacted factor. He has presented research findings in international conference held at Switzerland, World Immune Regulation Meeting 7, 2013, Germany 16th Joint Meeting 2012, GRK 1167 retreats in 2014. Sir is a resource, served as a resource person for several international conferences. Is having collaboration with scientists and eminent personalities from India and various countries. Presently, Sir is working as a postdoctoral researcher at Magdeburg, Germany. With this brief introduction, I welcome you, sir. I hope participants will enjoy this session. Thank you. Over to our convener, madam.
Thank you, sir. Thank you for your introduction to today's speaker. Now I would like to invite today's speaker, Dr. R. Amar Varma Garu, to deliver the lecture. Over to Amar Varma, sir.
Yep.
Sir.
Yeah. So am I audible now?
Yes, sir.
Yeah, right. Okay.
Yes, sir.
So
Yes, sir.
yeah, thank you, Dr. Jawahar Babu, for this kind introduction. So just before beginning my talk, am I audible?
Yes, sir. Yes, sir. You're audible, sir.
Yeah, right. So just a disclaimer before. No connection between my talk and to the title I have given there on the slide. So just I have chosen this. There should be some. So if you find. So let's begin the session. So, okay.
Sir, you are not audible, sir.
So, am I audible now?
Yeah. Yes,
now
yeah.
it is clear, sir. Right.
So yeah. So what I just told before was there is no connection between my title of the talk and to the content which I'm going to present. Hello? Am I audible?
Yes, sir.
Yeah. So, okay, so let's begin the talk. So normally we think that immune system becomes activated whenever it sees some. So, but it's not like that. Immune system becomes activated upon tissue damage. Tissue damage here, you see that it induces or activates immune system. Well, immune system per se, I mean, it's a part of host. So very disturbance here. So I don't know why this happens suddenly, but okay. So immune system is just a part of host defenses, but for simplification, we use the term immune system because it's just a part of host defense strategies. So upon tissue damage, so immune system resolves the inflammation by two strategies. One is, traditionally we think that immune system uses resistance strategy. So where immune system tries to kill the infectious agent and clear the inflammation. However, immune system also employs another strategy called tolerance. So tolerance is different from resistance. In resistance, the immune system kills the infectious agent. In case of tolerance, it only decreases the pathogen number while inhibiting the negative cost of the pathogen burden. Because when immune system tries to kill the infectious, hello? Yeah, there will also be collateral damage, but in case of tolerance, there will be no collateral damage because of the tolerance mechanism. It only decreases the pathogen burden or negative effect of the pathogen burden. So, however, there are certain pharmacological inhibitors which you may very well aware of. So, to dampen the immune response, one is the traditionally used non-steroidal anti-inflammatory drugs, and the second one is steroidal anti-inflammatory drugs. Non-steroidal anti-inflammatory drugs are, we use them day to day. For example, acetaminophen or diclofenac sodium or some other whatever. So aspirin, for example. So all these non-steroidal anti-inflammatory drugs, they inhibit immune response by inhibiting an enzyme called cyclooxygenase 1 inhibitors. You might know very well. In case of steroidal anti-inflammatory drugs, for example, corticosteroids, which are secreted by adrenal glands. So they basically shut off the entire immune system. So basically they inhibit the migration of leukocytes to the site of inflammation or infection sites. They also inhibit the gene programmings inside the lymphocytes, for example. Yeah? So that these lymphocytes or other immune cells cannot secrete inflammatory mediators, pro-inflammatory mediators to induce inflammation. I think you might have remember those inflammatory mediators or inflammatory cytokines from previous talks. For example, interleukin-1 or interleukin-6 and TNF-alpha, interferon, gamma, interferon, and so on. For example, interleukin-1 and interleukin-6, they are called endogenous pyrogens. They're primarily produced by liver, and they reach, for example, interleukin-1, they reach hypothalamus situated somewhere in our brain, and they disrupts the temperature regulatory mechanisms. The non-steroidal anti-inflammatory drugs, they stop the production of interleukin-1, and that's how they decrease the inflammation, because there is no interleukin-1 production or other inflammatory cytokines. Okay, so majority of all these inflammatory cytokines are produced by the subsets of leukocytes called lymphocytes, in principle by T lymphocytes. Okay, so let's look at the importance of T lymphocytes. So assume that this is a naive or virgin CD4 positive T cell. Naive, why naive CD4 positive T cell? Why not other compartments of CD4 positive T cells? Because naive T cells are important in recognizing new infectious agents or new antigens. Whenever, so all other T cells in a blood, they are not so important in recognizing the new agent. So because our novel coronavirus is new infectious agent, we have to talk about naive CD4 T cells. So CD4 positive T cells, they express a T cell receptor. So triggering this T cell receptor leads to the proliferation of naive, where is my cursor? Yeah, naive CD4 positive T cells into a subset of a clone of CD4 positive T cells. And from there, these clone of proliferated CD4 positive T cells can participate in different kinds of immune responses. For example, type 1 or type 2, type 3 or type 4, by secreting several cytokines. For example, type 1 immune responses is mediated by T helper type 1 cells. They principally secrete interferon gamma, interleukin 2, TNF alpha, tumor necrosis factor alpha. Likewise, type 2 responses, immune responses are mediated by Th2. They secrete interleukin 4, 5, 6, 7, 13, and so on. You might very well know about allergic responses, right? So those allergic responses are mediated by Th2 type cells. Okay? Next, type 3 immune responses are mediated by T helper 17 cells. So they principally secrete interleukin 17, 6, and interleukin 23. So these subset of T helper cells are mainly involved in protection against um, uh, uh, fungus. So antifungal responses are barrier protection. Okay, for example, many boys, some boys have soft hands. Perhaps if they go to gym and do some kind of exercises, you may see some kind of fibrotic lesions, thickening of the skin, right? So this thickening is mediated by the cytokines secreted by T helper 17 cells. So immune system thinks that there is a constant damage to the skin. So somehow I have to protect this damage, and that's therefore immune system it somehow thickens the skin. Literally everything whatever happens in our body, there will be immune system doing the repair. And finally, type 4 immune response is mediated by regulatory T cells. They principally secrete interleukin 10 and TGF beta. So the importance of T regulatory cells, as already mentioned by Sivajyoti madam. So they suppress or they regulate all these three kinds of immune responses. So if you remove this compartment, Treg compartment, from the immune system or from the body, then our immune system goes haywire and it produces hyperinflammatory conditions. Okay, right. So now what is the context of talking about T lymphocytes, and we will discuss in the following slides. So what happens when a T cell is triggered? So it leads to the activation of different intracellular signaling events. This slide is very important for you. So from here, it's mediated by several intracellular signaling molecules, all leads to the activation of three important transcription factors in naive CD4 positive T cells. So all these three transcription factors leads to the production of microRNAs, CD69, CD25. These two things are the surface activation markers. If you want to know whether a T cell is activated or not, you can just check the presence of CD69 and CD25 on the surface of T cells. And then these three transcription factors also involved in the proliferation of T cells and followed by the secretion of cytokines. Okay, right. Now, you already see that T cells, they secrete microRNAs. Let's look at the role of microRNAs and the context of this talk. So if you remember, I think in March or April, I don't remember exactly, that a research group from the Department of Biochemistry in New Delhi, they found that the SARS virus that is present in Indian subcontinent had a mutation in its genome. So because of this mutation, the SARS-CoV-2 virus is less pathogenic. That's what they found. And also they also discovered that there are several microRNAs in Indian can target the structural and non-structural components of SARS-CoV-2. For example, this is HSA, means Homo sapiens. MicroRNA 126, it targets nucleocapsid. And HSA, microRNA 23B, it targets spike glycoprotein, and so on. However, interestingly, here, this graph shows where exactly the mutation has occurred. So it's the the mutation occurred in the spike glycoprotein. If you remember from the previous talks, the SARS-CoV-2 virus uses this spike glycoprotein to bind the angiotensin converting enzyme receptor on host cells. So that's the spike glycoprotein location, genomic location is somewhere there in its genome. And exactly at 25,351, is it right? Yeah, 350, somewhere there. The nucleotide cytosine, it was mutated to thymine because of this reason. The spike glycoprotein, in the spike glycoprotein, because at the 930 position, instead of alanine, so valine has been substituted. So because of this reason, the virus cannot properly bind to the to its receptor. So because that's why it became less pathogenic. Okay, now let's look at what are microRNAs and how they are produced. MicroRNAs are already introduced by Ms. Sivajyoti ma'am. So they're a class of small RNAs, approximately 22 nucleotide in length. So they regulate gene expression in principle. So because of this, they can regulate different aspects of cell biology, because, for example, development, cell cycle, and homeostasis. And because of this reason, they can also be involved in multiple, they can be used, they can be involved in multiple sclerosis or atherosclerosis, diabetes, tumors, etc. So let's look at how they are produced inside the cells. So within the cell nucleus, RNA polymerase II transcribes the intronic sequences. We have in the DNA, we have two sequences, two regions. One is exons and one is introns. So these RNA polymerase II transcribes intron, and from introns, primary microRNAs are transcribed as long primary microRNA transcripts. From there, they are initially processed into 70 nucleotide length pre-microRNAs. And from there, from the nucleus, they are transported into the cytoplasm, where these primary microRNAs are further processed into small 22 nucleotide length microRNA duplex. From this duplex, only one strand will be loaded into the so-called RNA-induced silencing complex. Within this RNA risk complex, the microRNA targets it target genes. So if there is a perfect complementarity between the microRNA and a messenger RNA, so this leads to the degradation of target mRNA. In contrast, if there is an imperfect base pairing between microRNA and target messenger RNA, it leads to translational block. So the summary is either the microRNA can completely degrade the target messenger RNA and thereby decreases the translation, or it can imperfectly, or it can block the protein expression. Okay. So now, what is the role of microRNAs in T cells? You remember this slide before from this previous slide. So, microRNAs can regulate each and every step of T cell biology. For example, so microRNA 182, it inhibits the proliferation. MicroRNA 146A inhibits the development of Th1 subset, or microRNA 155, it promotes Th2 subset, or microRNA 155 promotes Treg compartment, and so on. So, well, I think, okay. So during the course of immune response or T cell responses, there are two phases in T cells. One is the initial phase of T cell responses. This slide is important. We will discuss about the importance of this slide in later. So there is an initial phase which is exclusively mediated by TCR. And then when cytokines are produced, so the later phase is induced. And during this phase, the T cells do not require or do not much depend on the signals derived from TCR. So, well, a part of my project was to identify new microRNAs that can regulate T cell activation. So, to identify which microRNAs are regulating T cell activation, so I did a microRNA array screen. So, I stimulated T cells and sent the samples for a microarray analysis. From this, I have focused on a few, I have selected few microRNA candidates whose expression is above 2 folds. They are microRNA 20A, 29A, 20B, 20A, 29C, 30E, 142, and 21. Of these 6 or 7 candidates, I focused my research on microRNA 20A because what I found from the real-time PCR is that only microRNA 20A was induced in the initial phase of T cell activation, that is 2 hours. So because as I said as I showed in my previous slide, the initial phase, which is mediated by TCR, does not last longer than, for example, 15 hours. So, yeah. So only microRNA 20A was induced upon 2 hours of T cell stimulation. So therefore, I selected microRNA 20A. All others are not so well induced. So why only microRNA 20A is, so microRNA 20A belongs to a big cluster called microRNA 1792 cluster. And this cluster is frequently overexpressed in hematopoietic malignancies and solid tumors. You may take any cancer patient or tumor or malignant patient. So they will all show the increased expression of the members of microRNA 1792 cluster. If you delete or if you overexpress this cluster, what happens is in mice, so mice suffers with severe lymphoproliferative disorder and also acquires autoimmunity. And if you delete this cluster in mice, it is lethal to mice. Mice dies prematurely, okay? And it was shown at the time that microRNA 20A was downregulated in multiple sclerotic patients, which is multiple sclerosis is a kind of autoimmune disorder. Okay, if microRNA 20 is, if multiple sclerotic patients show decreased expression of microRNA 20A, what does it tell? So an increase in microRNA 20A may somehow ameliorates the disease condition. And to understand this, if that is the case, so I induced experimental autoimmune encephalitis condition in mice and checked whether this autoimmune condition also decreases microRNA 20A. And as you can see here already, that microRNA 20A levels are significantly decreased in EAE mice compared to wild type mice, right? That means that microRNA 20A plays a role in the disease pathology of multiple sclerosis. So now my task is to understand how microRNA 20A regulates T cell activation. So, and to understand, and before doing that, I first checked whether how fast the microRNA 20A is synthesized and who are regulating the expression of microRNA 20A. As I showed that there are 3, the transcriptional activation of T cells. So they are NF-kappa B, AP1, and NFAT. So first, to understand this, so first I stimulated T cells, and you can see here this is the control cells, and these are the T cell stimulated cells without any treatments. So you see that a nice induction of microRNA 20A precursors upon 4 hours of T cell stimulation, which means that stimulation of T cells is triggering microRNA expression, which means that microRNA has a role in regulating T cell activation or signaling events. So to understand which transcription factor is regulating, I first block NF-kappa B pathway by a pharmacological agent called IKK7. It is a pharmacological inhibitor. So yeah, when you treat T cells with this inhibitor and then stimulate the T cells and compare the expression, what happens is, right, NF-kappa B is regulating, somehow the absence, the NF-kappa B inhibition is also inhibiting the expression of microRNA 20A. So likewise, if I inhibit RAS-ERK pathway and AP1 activation by using the pharmacological inhibitor U0126, what happens is, so inhibition of AP1 activation also significantly decreased microRNA 20A expression. Likewise, I also checked if NFAT has any role. So NFAT is activated by calcium influx inside the cell. If there is more calcium, so that leads to the activation of NFAT, and therefore it leads to more cytokine production, microRNA production. So I blocked calcium influxes in T cells by using EGTA. You might have very well know about this compound. So if I block NFAT activation, sorry. So oops, suddenly all microRNA is gone, which means that microRNA 20A expression is exclusively dependent on NFAT completely and to some extent on AP1 and NF-kappa B. So now I wanted to check what is the role of microRNA 20A, what it is doing inside the T cells. So to understand this, I overexpressed microRNA 20A in T cells and checked how it affects cytokine production in T cells. So these are the cytokine profile I have done. So out of these all cytokine profiling, for example, interleukin 2, interleukin 4, 6, interleukin 8, TGF beta, TNF alpha, interferon gamma, and interleukin 10. So you remember from the previous talks that in COVID-19 patient, there is cytokine storm, right? These cytokine storm is mediated by all these pro-inflammatory mediators. However, microRNA 20A was only has an effect in the production of interleukin 2, interleukin 4, interleukin 6, and interleukin 8, and interleukin 10. So the red bar is overexpressed T cells, and the blank bar is wild or untreated T cells. Compared to untreated T cells, so microRNA 20A overexpression is decreasing interleukin 10, interleukin 2, and so on, which is in line with the initially observed data that microRNA 20A somehow downregulates or inhibits T cell activation, and so it also play a role in controlling the inflammation, for example, multiple sclerosis. So because there are no enough cytokines, pro-inflammatory cytokines to mediate inflammation. So which means that microRNA 20A is negatively regulating T cell activation. Okay, so now you know this slide from the previous talks that SARS virus enters into the lungs and binds to the angiotensin converting receptor and activating alveolar epithelial cells and monocytes, macrophages, T cells, and so on, producing pro-inflammatory cytokines, and also the activation of B cells and so on. So what happens inside is, so that's the infected T cell, infected lung epithelial cell. So because of this infection, the infected cell undergoes necrosis, and because of this necrosis, cell releases of certain danger molecules, like tissue damage. And the tissue damage activates the immune system. For example, here the first responders are macrophages. The macrophages then produce several mediators that activates polymorphonuclear leukocytes or neutrophils that produce reactive oxygen species, peroxidases, matrix metalloproteases. So all these mediators, for example, reactive oxygen species are contributing to severe oxidative stress inside the body, and matrix metalloproteases, they break the collagen, the connective tissue, and because of the production of interleukin 6, it also activates B cells to produce antibodies, and finally, it also activates T cells. For example, in case here, the CD8 positive T cell, which is secreting interferon gamma, interleukin 6 perforins, and granzymes. These cytokines, they target the infected cell and destroy the virus inside. Okay. So, however, there are two phases in viral infection. One is acute or acute viral infection, and the second one is chronic. So, in case of acute viral infection, the infection is controlled by controlled in the early stages by innate immune cells and later by the adaptive immune system. They together somehow control the viral load. So therefore, patient will be recovered. However, in case of chronic viral infections, so immune system will be continually triggered. So therefore, you have so much of cytokine burden inside this, inside the body, but this blue line, adaptive immune system, will not be as active as it was in acute viral infection. This is because the T cells, when they are repeatedly activated, when they are repeatedly poked by the triggers, antigenic triggers, they somehow undergo T cell exhaustion. So they no longer perform their functions. And we need T cells to clear the viral infection from the body. So this is the reason why the chronically or severely infected patients do not very unlikely recover from the infection, because there is no T cells to clear the infection. So here, for example, the CD8 positive T cell is required to attack the infected cell. So because of the T cell exhaustion, there will also be lymphopenia and immunosuppression. So this feature is very frequently observed in those patients who die from COVID-19. So this is because it's not yet so clear why the lymphopenia means absence of T cells. So all the primary lymphocytes and their numbers will be decreased. And also the Treg compartment, which I told in the previous slides, Treg's compartment is important for the suppression of immune system. So the hallmark feature of the COVID-19 patients is there is also a decreased Treg compartment. Because of the loss of Treg compartment, you see a lot of cytokine storm inside the patients. So now, how to manipulate the immune response because of from this cytokine storm is, so there are several strategies. For example, so once the virus enters inside the T cell, when it enters into the target cells, so every cell is equipped with certain immune recognition. Yeah, equipped with certain immune sensors. For example, here the endosomal RNA sensor called which secretes interferons and interferon regulatory factors, and they secrete interferon gammas. And there are also cytosolic RNA sensors, because this is RNA virus. So the RNA virus, the intracellular RNA will be recognized by some other intracellular immune sensors. And all these immune cells sensors, they trigger the cells to produce more cytokines. So if you somehow block these sensors from recognizing the infectious agents, so somehow you also dampen down the cytokine storm. Next, so in addition, so to effectively maintain T cell populations, so you can also use certain cytokine inhibitors. For example, interleukin-6 inhibitors and interleukin-1 inhibitors. These are, because this is important, because in the initial phase, it is mediated by innate immune system, which secretes already certain cytokines. And later on, adaptive immune system takes over, and it also is secreting more cytokines. And because of this, more inflammation is there. And if the production is continued, the T cells will undergo exhaustion. Therefore, if you use in a temporal manner the inhibitors, interleukin-6 or interleukin-1 inhibitors, using corticosteroids, for example, that's the reason why people were asking the questions about the using of dexamethasone. So this leads to the damping down of T cells for short times. So therefore, they won't undergo exhaustion, and they will be ready to take over the next phase when the corticosteroid effect is gone. So they will be again, they are strong enough to combat the virus. In addition, you can also use interleukin 7 or N-acetylcysteine. So N-acetylcysteine so neutralizes the oxidative damage, and interleukin 7 is important for the homeostasis of T cells, so that T cells do not undergo T cell exhaustion and followed by T cell deletion. So this interleukin 17 maintains T cell homeostasis. Therefore, T cell responses will be sustained and they won't undergo exhaustion. That's how you can improve T cell responses. Likewise, you can also use convalescent plasma therapy, so which selectively binds to the virus particle and somehow clears the infection, and therefore also decrease the burden on the T lymphocytes. So, and this slide tells the other options of blocking the cytokine storm. For example, tocilizumab, which is an interleukin 6 inhibitor. So, and also all other ruxolitinib, whatever. And this is an pharmacological agent to block JAK-STAT pathway, and also infliximab, which is an antibody that binds to the TNF, tumor necrosis factor alpha. So all together inhibit cytokine storm and therefore inhibit immunopathology inside the COVID-19 patients, and therefore decrease the pathogenesis of caused by the virus. And there are also certain other therapeutic drugs against COVID-19. So if you remember from the previous talk that there are certain structural and non-structural protein on the coronavirus. They are E2-glycoprotein, envelope protein, ORF1 or ORF3 and ORF10, and ORF7 and ORF8. So all these molecules, they bind to heme molecules and dissociate iron from these heme molecules. And once the iron is dissociated from the porphyrin, then these viral components bind to porphyrins. So already in yesterday's talk, we so the speaker told that hemoglobin is composed of heme, and which is held by two alpha hemoglobin chains and two beta globin chains. So it's like a bomb in a box held by four persons. And then these viral protein arrive to this hemoglobin molecule, and they pull out these chains from this heme compartment, and then they open the box. So the iron molecule will be escaped and then binds to the porphyrin. So there is investigation is still going on how virus enters into the cells. So normally we know that SARS-CoV-2 binds to the ACE receptor, but some studies says that the interaction between spike glycoprotein and the ACE2 receptor is very weak. So that they think that it's not the correct. This does not facilitate virus entry inside the T cells, and there will be some other mechanisms. So the other mechanism is that once these molecules bind to the porphyrins have the capacity to enter inside the cells. So they think that that's the reason why they think that why envelope proteins or virus targets hemoglobin molecules. So using these porphyrins, they enter inside the T cells, but there is the iron is left over. So this iron is a very pro-inflammatory agent. So for example, in our body, for example, hemoglobin is present in RBC. So when RBC life cycle is over, so RBC will be degraded, and the heme component will be bind to the heme binding proteins, and they transport it to the liver. So in the liver, the heme is dissociated into, degraded into, first into biliverdin and then bilirubin. And then iron is again recycled into the hemoglobin, right? So now, so we talk about now chloroquine, how chloroquine is used or inhibits viral replication. So chloroquine is first derived from the compound called quinine, so which is extracted from the cinchona bark. And you see this structure of quinine compound, and adding chlorine molecule to the quinine leads to the chloroquine. And if you add hydroxy group to the chloroquine, it becomes hydroxychloroquine. So that chloroquine binds the E2 glycoprotein and over of 8, so that these two viral proteins cannot bind to hemoglobin. So here, for example, this is the red thing here. You see that? That's the chloroquine molecule. So once it bounds to the pocket domain of these viral proteins, so it cannot any longer bind to the hemoglobin. And it also decreases acidic environment in the intracellular compartments. You see that in case of viral replication, so viral components are assembled into the endosomes. Endosomal compartment is always acidic. Without this acidic environment, the protein molecules are not properly conformed. So when you take chloroquine, it decreases the acidic environment. Therefore, viral assembly is also decreased. And likewise, it also inhibits TLR activation. So the TLR activations are the immune recognition molecules or receptors on cells. So constant triggering of these molecules or immune sensors leads to the production of cytokines and leading to the cytokine storm. What chloroquine does in some way is it also inhibits TLR activation and therefore decreased cytokine storm. And the recent one, very latest one, is Fabiflu. This Fabiflu is approved in 2014 in Japan to treat influenza viral infections. So the compound is called flavipiravir. So it also binds to the ORF7 and ORF1 proteins of virus. So therefore, the viral entry will be inhibited. I said that already, that these ORF, they use porphyrins in order to enter inside the cells, right? So, and they also inhibit viral RNA polymerase activity inside the cells. So so to make RNA copies and viral copies, the viral RNA has to be multiplied. So this Fabiflu also inhibits RNA polymerase activity. But however, the company claims that this can be used only to treat mild to moderate infections. But you have to remember that 80% of the COVID-19 coronavirus infections are mild. So check before using Fabiflu. And there is another drug which is now under a randomized placebo-controlled phase 2 clinical trial, which is called Camostat mesylate. So this compound inhibits the binding of virus to the ACE2 receptor. So which is, this trial will be over. They say that the trial will be over in December. And in addition, there is an independent also clinical trial is going on, that is Camostat mesylate and chloroquine. I think they said that they start this clinical trial in this month, June. This is June, right? So June, yeah. So in addition, so there are some other therapeutic drugs which I'm not talking about here. Some other are, I think in 2016 or 17 or 18, 2018, I think. Yeah. So an Indian researcher in Yale University, so she accidentally discovered that neomycin, which is an antibacterial, if you use this neomycin drops intranasally, so it inhibited influenza infection. So since, I mean, the mechanism mode of action is, so the neomycin drops. So even though it is a bacterial antibiotic, it is activating the lung epithelium, the immune mechanisms of alveolar epithelium. So, well, here also coronavirus also attacks alveolar epithelial in the beginning, and then therefore it enters into the lungs. So perhaps neomycin can also be used, but I mean, one can try, I mean, test in clinical trials whether neomycin can be used. Already, I also told you just before that chloroquine can be used to treat the COVID-19 patients. And there is also, I mean, the homeopathic intervention, therapeutic intervention is already started with the discovery of, I mean, with chloroquine, because the Hahnemann, he discovered that when you use chloroquine, when you give to healthy individuals, so what he discovered, what he observed was it induces malaria-like symptoms. And finally, a patient will be healed from malaria. So based on this, because chloroquine is also used to treat COVID-19 patients, one may also try to test whether homeopathic formulations of chloroquine can also be used to treat COVID-19. And also there is arsenicum album. Yesterday we discussed this. So arsenicum album, just like neomycin, it also activates the immune mechanism, immune defense strategies of alveolar epithelial cells. You may wonder that we have designated immune cells. Why again other cells have to perform the same function? But we have to remember that every cell tries to live, tries to survive. It's the survival of existence, right? So every cell tries to survive. So therefore, it acquires some resistance strategies against infectious agents. So when you use these arsenicum album or neomycin, so they activate those hidden immune defense strategies in these epithelial cells. So that's about the therapeutic strategies. So I wrote 2 articles in local newspapers. So one is on April 23rd. So in this paper, I talked all those treatment strategies. And in the second article, I talked about how effective the lockdown was and the importance of and the benefits of herd immunity, which was published in 7th May. Students who are interested can check these articles. And now I close this session by thanking you. So this slide was, I taken this shot in Davos Congress. So you see this. So she's Polly Matzinger. So much of my thoughts were stimulated by Polly's ideas. So we think that immune system is activated by the foreign agents, but she suggested that no, it's not like that. It's because immune system gets activated because of tissue damage. Yeah, and that was, I think, many Telugu people will associate with this name. Particularly Andhra people. Every year, our government used to go to Davos to do some things in March. So yeah, that's Davos for you. And I close this session, and I'm open for the discussion.
Thank you, sir, for your awesome talk. Let us have a questionnaire session now. Dear participants, if you want to ask any question, please type in the chat box. Sir, there is a question for you.
Yeah.
Can Fabiflu will be useful to patients below 18 years? Fabiflu.
Yeah.
Whether it is used to, sir, can Fabiflu will be useful to patients below 18 years or not? Below 18 years.
The company says that from 20 years to, I think, above 20 years, 20 years above. I'm not sure. So this has to be directed by the physician. But I think in principle, yes, I think they can be. But I'm not sure. What company says is about 20 years.
Thank you, sir.
Yep.
So another question, sir.
Yep.
What are the functions of different ORFs in COVID-19?
What are the functions? They are non-structural proteins. So perhaps they are used to assemble. They will be used to assemble the viral components. But I'm not sure.
if you another question, sir. If you suppress the T cells, it affects the immune response, or it affect increases the chances of COVID-19 disease. Suppression of the T cells, how affect on the COVID-19 disease?
So that's the problem with the T cell responses. So because there is continual triggering of inflammation, so T cell undergoes exhaustion. So you need to suppress T cell responses in a timely manner, not completely. You observe. So T cell responses start after 4 to 7 days. You give them 3 days of time, and then give a shot of corticosteroid or immunosuppressive agents. Wait for 1 day, and then again remove the break. So allow the T cells perform their actions. Again, apply break. And then allow intervals, frequent intervals, so that if you allow T cells, they run, and then they exhaust. So suddenly there is a drop down in immune functions, and which allows the virus to take over.
Okay.
Yeah.
One question, sir. Can you give the T cells to enhance the patient immunity?
Mm-hmm.
Is it the COVID patient is having the deficiency of the immune system? If you are boost of the immune system, you are giving T cells. Is it correct, or
adaptive T cell therapy? Adaptive T cell therapy, but in principle you can do that, but only when there is lymphopenic conditions. You can time to time you can check the T cell number, like you do the test for HIV patients. If there is a drop down in T cell number, then you can adaptively transfer T cells. But the care has to be taken, because just mere transferring of T cells won't help. We need antigen specific T cells. So what we can do is, so you can culture antigen specific T cells, and then you can transfer them adaptively. But again, it's a double-edged sword. Too much of antigen specific T cells will lead to too much production of cytokines, leading to the cytokine storm. Yeah, I mean, in principle, you can do with the antigen specific T cells.
One question by the participant.
Yep.
Is it microRNA therapy can be used to treat the COVID-19 disease?
Yeah. So you know the mechanisms of microRNA mediated regulation. So microRNA, so it just see if there is any complementarity binding targets. So if there is a perfect complementarity or it just binds. So likewise, you can synthesize the viral RNA binding microRNA. That means microRNA that specifically binds to viral RNA. So you take these, you synthesize, you make these microRNA, put them in exosomes, and give them intranasally, inhalation. So then these exosomes will bind to the alveolar epithelial cells. They go inside the cells, and if there is virus RNA, it just go there and binds to the RNA and degrades it. Therefore, from this degraded RNA, so viral proteins cannot be made, and therefore viral load will be simultaneously reduced. That's how it works. And you can also stabilize microRNA so that it stays longer inside the cells. Normally microRNAs they do not stay forever, right? They have certain half-life. So you can increase the half-life because you are chemically synthesizing. So you can induce some alterations so that it will be stable for longer times. Put them in exosomes, like lipid molecules, and then you can just inhale. Yeah, it works. Yeah. I mean, people are thinking about these therapeutic choice.
One question asked by the participant, sir.
Yeah.
What is the scope of nanobiotechnology for facing this COVID-19 pandemic? How nanobiotechnology can use it to this pandemic situation? Or
well, I do not know exactly the scope of nanotechnology itself. So if you make small bots, which can specifically recognize the target cells or infected cells, and you can destroy them. Or, I mean, microRNA strategy is also a nanotechnology.
Yes.
Nanotechnology. You can make exosomes or liposomes, for example.
Yes, sir.
Yeah.
Yes. That's question X. Yes, yes.
Sir, there is one more question. Hydroxychloroquine is having several cardiac toxicity effects, but still so many physicians are recommended to the COVID-19 patients. Why they are recommending, sir?
So many what? They are recommending what?
Hydroxychloroquine drug is having cardiac toxicity effects, sir. It is showing so much of cardiotoxicity.
Yeah.
But still, why the physicians are referring the drug?
Well, I mean, I think it's yeah. But I think it's the question is somehow. I don't think physicians or doctors, medical doctors, will prescribe this hydroxychloroquine if they have any comorbid conditions. For example, if they have cardiac problems or diabetic conditions, I don't think they prescribe chloroquine. Yeah. I think it's only the otherwise healthy individuals only they prescribe the chloroquine. There are certain studies that chloroquine doing, inducing cardiac problems. But for others, I mean, it's okay. I mean, you have to understand that chloroquine is naturally occurring compound, so it has multiple effects. It's not chemically synthesized product, so that it specifically binds one particular molecule inside. Since it is a naturally occurring compound, it has diverse effects once it enters into the human body. Yeah.
What is the current status of coronavirus vaccine? One person asked like this.
Well, you know what? To make a vaccine, we need at least 10 years. That's the required time frame. But for Ebola virus, it took only 5 years because the Ebola virus was so pathogenic. It was because the pathogenesis is it destroys blood vessels. So on a war footage, they made the vaccine in 5 years. But now, the virus was only, the infection is started, I think, 6 months ago. I don't think, there are certain, for example, Oxford University tied up with some other company in India. So they are trying to make a vaccine, but we don't know how effective the vaccine was. So I think it takes some more time. It's too early to expect a vaccine at this moment.
So one participant, sir.
Yep.
What is the macrophage-based vaccine approach for COVID-19 immunotherapy?
So
macrophage.
Macrophage.
Yeah. If you are the vaccine is prepared based on macrophage approach for COVID-19 immunotherapy.
So based on macrophage.
Macrophage.
No, I don't think you can make. Well, you can train innate immunity, but I'm not sure because at the end you need cells that clear the virus infection. So all macrophages, so they activate other immune cells, but let me think. I'm not sure because it's so far all the vaccine strategies are. Hello?
Yes.
So for all the vaccine strategies are mainly based on so to provide long-lasting adaptive immune response or long-lasting antibody responses. And I think this is a new strategy to make virus based on trained innate immunity. But I don't know, I'm not sure how long this will last once you even though you make a successful vaccine, I'm not sure how long this will last. So it's new.
One last question, sir.
Yeah.
During peptide vaccine development, what type of proteins can be best candidates for peptide screening for COVID-19?
I mean, you have to screen for all those peptides that give a stronger immune response, or not even stronger, but long-lasting immune response. That's the key strategy, key factor.
Candidate.
Yeah, peptide candidate, which gives you long-lasting immune response. Because although all components of virus are antigenic, only few antigens can give long-lasting immune response.
Yeah. Sir, small question from my
Yeah,
side.
no problem.
Yeah, if you are using the diagnostic test, that RT-PCR is more sensitive compared to blood test.
Blood test. Okay, yeah, I see your point. So, well, I mean, RT-PCR. So, we don't know what we are going to test, what we are going to identify. If you want to test the presence of virus itself, it doesn't tell. It only tells if a person has a virus, but it does not tell the strength of the person, the immune. The immune status of the person. So, okay, it just tells the virus is present in this body or not. But if you use blood test and detect the antibodies, then this tells the immune status of the person, so that you can decide whether to send this person to the quarantine or suggest a home quarantine, home isolation. So I think blood checking, blood test will be better choice than RT-PCR. RT-PCR is more expensive compared to blood testing antibodies. You can just check antibodies and say, based on the titer, you can say that whether the person is responsive, resisting the infection or not.
Sir, is there any complication in plasma therapy other than the cost?
I think plasma therapy, I think there won't be any complications, because you use only plasma components. So they bind if there is virus. Otherwise, after their half-life is over, they will be removed or eliminated from the circulation.
Thank you, sir.
Yep.
Thank you, sir, for your wonderful explanation.
Yep.
Your lecture is very informative, thought-provoking, and fantabulous, sir. You have analyzed all the basic areas of immune response, like signaling events to trigger the TCR, mutations in Indian coronaviruses, and role of microRNAs in T cells, microarrays, viral infections, manipulations of immune responses in COVID-19. And one thing, sir, really I admired a lot regarding one topic, that is therapeutic drugs against COVID-19. You have covered the recent drugs also, like Fabiflu. I have clarified my doubts here. All were really fantabulous explanations, sir, for all the questions. And the participants also feel very happily regarding your explanation and regarding your presentation, sir. We are very fortunate to have a speaker like you for this online international webinar, sir. I hope all the faculty and student participants have gained excellent knowledge from today's session. Now I request our Honorable Secretary and Correspondent, Sir, Dr. S. Madhusudan Rao Garu, a man with a great vision, highly knowledgeable, focused, a strategic leader, and have a passion for excellence. Sir, please express your views and opinions on today's session. Over to Madhusudan Rao, sir.
Okay, am I audible?
Yes, sir. Yes, sir.
Yes, sir. Yes, sir.
Good afternoon to all. Sir, Dr. Verma, very good afternoon, sir.
Good afternoon, sir.
I'm a fan of yours. That's why I came to listen to your lecture, actually.
Thank you, sir.
As you have shown, I have followed your 2 articles in Enadu sometime back. And I'm astonished about your Telugu, which you have preserved even after going to Germany for a long time. Both of them are very wonderful articles. And having seen your biodata, it's mind-blowing and rather.
Thank you, sir.
In a matter of span of 10 years, you have done all the technical procedures that were present in your institute, which is one of the best in the world, probably, instead of molecular and clinical immunology. it has forced me to go through your website also. And I am really proud that you are a Telugu man, our own man, and also you graduated from a college from Ongole and Kurnool.
Yeah. Oh, yes, sir.
Probably our faculty, Dr. Johar, did not tell this. And from there, you have jump-started your career. That's really wonderful.
Yes.
Ireland, from there to Germany, and doing all sorts of research, right from isolation to gene expression to stimulation of T cells, measuring of calcium influx in cells, detection of cytokines, measuring activation, proliferation, inhibition and exhaustion markers, pharmacological treatment of T and B cells, intracellular staining, measuring apoptosis. I was reminded by this when I was in first year of my MBBS. My professor used to tell me, apoptosis is a civil procedure, but necrosis is a criminal procedure, he used to tell me. Of course, really, the work you have done in both ways is really mind-blowing. A lot of project work also you have done. What all you have explained, you have done probably in your project number 1 when you have started your PhD career.
Yep.
I feel. And we are really fortunate, sir. How I wish you are in India and you came to our college once. Please, we would like you to take the very first opportunity to visit our college and address our faculty and students. And my own dream is that biotechnology here in our college should be a branch-based, full-fledged branch. Given all the other prospects and though it's a growing subject, we've been hearing that it's growing since a long time.
Yep.
But there should be a for biotechnology because it's a important place and valid place for biotechnology. So schools must be encouraged to do a lot of experiments and a lot of procedures in biotechnology, and we wish both of our faculty members, Ms. Sivajyoti, who is going to be a doctor shortly, and Dr. Johar, both are very enthusiastic and they need your help from time to time, sir. I request you to kindly be accessible to them.
Sure, no problem.
Thank you so much. And last compliment from me, all the 3 speakers, right from Dr. Anna to Srinivas Garu to you are, I can label you as awesome threesome. Because all the lectures are so interrelated and so coordinated, without any effort. So all the 3 are very good, and we seek your help and guidance.
Yeah.
Thank you.
Pleasure, sir.
Thank you, sir. Thank you.
Thank you, sir, for your good opinion on today's session. Now I request our Vice Principal, Madam Dr. A. Sunita Garu, to express her views and opinions on today's session. Over to Sunita, Madam. Ma'am.
Thank you, Shruti. Verma, sir.
Yes, sir, madam.
Yeah, I'm very happy to attend this session, sir. It is really inspiring. Apart from today's session, I would like to request you to share your experience regarding about the postdoctoral fellowship, because many of the youngsters are attending this session, sir. Now the students, PG students and faculty members. So you please share your experience, how to approach, how to apply for postdoctoral fellowship.
So, yeah, so am I audible?
Yes, sir.
Yeah, so I think if you finish a doctoral, if you have a doctoral degree, then you can directly apply to the laboratories who you have an interest. So you can check online. So for example, in Germany, or you can also alternatively check by different job advertisement websites, for example, Nature Jobs or Science Jobs, or you can directly check your area of interest and plus country. So for example, let's say T-cell research, Germany, T-cell research, America, or in more specifically, for example, NF-kappa B, for example, or AP1 or whatever, or the molecule, target molecule, or signaling molecule, for instance, or autophagy, for example, the mechanism, or apoptosis, plus, kind of in such a way you can directly search for the labs and directly write to the PI, because they like to receive applications. So do not hesitate to write them and ask if they have any. Please read their research before applying, and try to make your own research statement or own hypothesis. So this and this I want to do in your laboratory. So if you have any opportunities, so you can also say that I can also wait. If at this moment, if there is no funding is available, I can wait. Or you can also say that this and this is my research interest, and I'm also open to whatever research that is currently going on in your laboratory. So you show some interest. So, and then also check on jobs advertisementing websites. So that's the way only. Or if you meet the speakers, international speakers in conferences, for example, live conferences, you can directly approach them. So that's another way of obtaining research opportunity.
Is there any open postdoctoral notifications?
Madam, I cannot properly hear your voice somehow.
Is there any open postdoctoral fellowship notifications? Not
Yeah, they are all open, ma'am. They are all open. They are all open.
Frequently, which websites we have to continuously
Madam, so,
monitoring?
madam, so there was some small disturbance. So once again, please.
What are the specific websites we have to continuously monitoring?
Yeah. Yes. Yeah. So one is I can type here, for example. So can you? Why it went some privately, but I'm sorry. So it's not going direct to everyone. I typed just a message here, but it went to someone. So it's not to everyone. I cannot see a message to everyone. So I can say nature jobs, naturejobs.com, or it's for everywhere. So you can say, so it opens a web page. You can type, for example, immunology and the country, or the topic and the country, and also jobs.ac.uk. jobs.ac.uk. So this is only specific for UK jobs. There are several categories. You can select biological sciences, and well, it's there. So yeah, jobs.is. Yeah, I can see now there. Yeah, and yeah, that's.
So the topic which we have selected for today's session is really impressive. The present pandemic situation, everyone have to concentrate and know much more about the T cells. T cells plays a pivotal role in pathogenicity. So I hope all the participants have acquired sufficient knowledge and can go for more research. COVID-19.
Yeah, thank you, ma'am.
Thank you, ma'am. Thank you, ma'am, for your good opinion on today's session. Now this is the time for proposing vote of thanks. On behalf of Hindu College of Pharmacy, I would like to take this opportunity to express my gratitude to today's speaker, Dr. Amarvarma Garu, for his excellent lecture. We are all inspired by your great lectures, sir. Thank you. I thank the management of Hindu College of Pharmacy for their continued support. I thank the principal, Dr. R. Govindarajan Garu, for his continuous encouragement. I thank the vice principal, Madam Dr. A. Sunita Garu, OSD Sitaramaya Garu, all HODs, fellow faculty members, technical and supporting staff. I especially thank to all participants for their active participation. Dear participants, we value your presence with us and look forward for continued support on tomorrow also. Tomorrow, the session will start sharply at 2:30 p.m. We have the lecture on innovation and incubation in biotechnology and pharma sector by Dr. S. Jahurullah Mohammed Sir, Chief Innovation Officer, Government of India. Dear participants, there is a small announcement for you. A feedback link is given in Zoom chat box. You can find it and fill it, submit within the stipulated time period. It is also given in the Telegram group. Thank you one and all. We are closing the session. Stay home, stay safe.
Thank you.